Hello, everyone, and welcome to Chapter 17. We'll do this in two parts. So, as we get started, just as a reminder, it's hard to beat a person who never gives up. Babe Ruth. So, don't give up. Never give up on me. All right, so here we go. What we're going to do, we're going to jump into this discussing some things. These are the things that we are required to discuss. What I do make sure to discuss is that I test on every chapter objective. So something from each one of these will be on the test for sure. There are some other things that I do test on that aren't on here, and you'll hear me mention that in my lectures just to kind of always let you know that I do test on these things. And for those who are watching this who aren't my students, just watching it because they came upon my YouTube channel, thank you for being here. So let's jump into this and really just differentiate senses. Now, some of this, if you had me for AMP1, we talked a little bit about general senses already. We talked about nociceptors, thermoceptors, tactile receptors, and we talked about proprioceptors. We talked about these in the brain and cranial nerves lecture where I discuss these different types of pain, temperature, touch, pressure, vibration, and proprioception things. Remember, pain, that's nociceptors, temperature, thermoceptors, touch, and pressure, vibration. These are special types of tactile receptors. Proprioception is a very special where the joints and body are in position in space, those kinds of things we briefly talked about. But these are general senses. They tell us general information. They don't have organs that are found in one place, like a pain receptor is found all over the body, but a smell receptor is found only in the nose. Vision receptors in the eyes, taste on the tongue and throat, balance in the inner ear, hearing in the inner ear. These are special senses. They are specially located in complex organs with specialized receptors in specialized places, found pretty much, they're found in the head. They're found all in the head. They're neurons. They're very specialized for that. Remember, we talked in AMP1. I said a neuron, a receptor, is only sensitive to one type of sensation, one type of stimulus. And whatever that is, that receptor is sensitive to, it's attached to a sensory neuron, and that sensory neuron only sends information from that receptor, one receptor to one sensory neuron. Remember, we talked about the labeled line principle. Now, the idea here is this is internal and external conditions of the body. So as we go through this, let's get the idea of the five basic senses that aren't things like touch. Touch is an example of a general sense, but these are other than your special senses, other than the touch and ones previously thought that we talked about. These, when we say, people say about the five senses, we talk about the five special senses. There are more senses than that. Olfaction, which is smell. This is one that is lost because of COVID. The assisting receptors that are there, the assisting cells, the special cells that assist on the side, supporting cells die off, and then you have lost your smell. Gustation is taste. I always like to say I like food with some gusto, with some taste to it. Spicy. I really love spicy. Vision, of course, that's seeing. Equilibrium, that's balance. And hearing, of course, we know what that is. Now, these all have the different things other than nociceptors, thermoceptors, tactile receptors, proprioceptors. They have different kinds. So these are specialized neurons, and we'll talk about their specialized cells as we go along. Now, the first one we want to talk about is olfaction. Olfaction is done by an organ called an olfactory organ, and these are up in the upper part of the nasal cavity. They're in the superior upper part of the nasal cavity, to be a little redundant there. And they have these two layers to them. Now, we're not going to sketch this. This is not something I spend a lot of time sketching, but I talk about olfaction. Olfactory receptors, they have these receptor cells and supporting cells. The supporting cells get damaged by COVID, and you can lose your sense of smell. The stem cells or basal cells that also regenerate the olfactory epithelium. So the receptor cells are part of this. And they're in the upper roof of the nasal cavity, And underneath this is a bunch of connective tissue and blood vessels and nerves called the lamina propria. Lamina propria is the underlayment that basically contains the glands that secrete water and make a pigmented mucus. The mucus can take on certain colors, so we know about that. Now, so as you can see, it's up here in the upper part of the nasal cavity, the pigmented epithelium, not pigmented epithelium, sorry, the olfactory epithelium. Pigmented epithelium is part of the retina. Me saying words again. And this olfactory nerve comes in, the olfactory bulb. We have branches of the neurons coming down, and what these neurons do is they emerge downwards through the olfactory organ, and they branch out their dendrites all along the surface of this. Here you can see the lamina propria with its gland rooted in here, and the olfactory glands secrete mucus that act as flypaper to trap odorant. Odorant are the molecules that we actually smell. Now, this is, to me, the really bizarre thing about smell. When you smell a flower, molecules that were in that flower come into the air. They are brought in. These substances being smelled, they are molecules that were once in that flower, and they bind to these olfactory cells, these receptor neurons, sensory neurons, specialized neurons, and they bind to it to stimulate it. And they're going to bind to proteins on the surface of it. So anything you smell, anything, molecules that were in that end up there in the olfactory epithelium. So even when you smell feces or anything like that, molecules of that were part of it are binding to these cells up in your upper roof of your nose. And that is always a little unnerving, isn't it? Now, how they work is we talk about G protein linked things before, and I said, now remember, G protein is going to be like your little brother or little sister, and you are, let's say in this case, you're the odorant, you're the molecule being smelled, the thing you smell, you're the odorant. And you snuck out of your house to go see a concert your parents said you couldn't go see. Now you try to sneak back in, and little brother, little sister, you're knocking on the window to see if they'll let you in the bedroom. Now they could let you in and be cool and let you in, and you get away with it. Or they could be like, no, stay out there and freeze. Or they could say, Mom, Dad, brother, sister snuck out of the house last night. Now, of course, it depends on what kind of little brother or little sister you have as to what's going to happen. It depends on the G protein, depends upon what kind of response. How will your body perceive it? What will the chemical, how will it be smelled? Things like that. So what you get, these odorants bind to these receptor proteins that are on these neurons and fire them in a certain way to set off a specific signal that these neurons will produce. And eventually, for you to be aware of it, it's got to get to the olfactory cortex of the temporal lobe for you to understand it. To get there, it takes the olfactory nerve, cranial nerve number 1. We did a cranial nerve review in my first lecture. I'm going to sip a little. This is my first recording post-COVID, and I'm going to sip some coffee while I'm doing it just to keep my voice going good. All right, so let's talk about a little bit of that. As I said, when these molecules bind to it, these chemicals bind and they generate a very specific set of signals that are there. And this signal being present, these depolarizations and action potentials create a unique signature that we would help recognize as that's what this chemical is. And they could do it by the odorant binds, and then it sets upon a cascade of events that activates the cell using G proteins. Here we have an odorant. It binds to a receptor. G proteins can get activated, and this sets a cascade of events that can stimulate the cell to fire and create that action potential that tells me what's going on. Now, there could be odorants that fire more easily, things that are very soluble, water and lipid soluble, can stimulate very easy. That means a lot of, if their solubility is good, they can penetrate the mucus and they can give you strong smells. Things that don't stimulate it that much give us very weak smells. There are some smells for us that are very strong. One of those is skunk stink. That is one we can detect at very small levels. Now, gustation is very similar. Instead of odorants, these are things that are in the food and drink that we ingest, things that I swallow. The taste is about the food we eat, the fluids we drink. There are a chemical cocktail in that stuff, and these gustatory receptors or taste receptors are on the tongue, on the upper part, superior part of the tongue. They're also down in the pharynx and larynx, but the most important ones, of course, on the tongue to tell us what's going on. These taste receptors get clustered, get clumped up into special cells that are epithelial cells, and those epithelial cells and taste receptors group together and form a taste bud. So a taste bud, as you can see, here's a taste bud. There are these epithelial cells and specialized neurons that all combine together into forming a taste bud. Epithelial cells, specialized taste cells, gustatory taste cells, all come together to create a special combination of what is called a taste bud. Now, these taste buds are found inside of lingual papilla, many of them. Now, there's one that has none. Filiform papilla. The filiform papilla, now I've checked, our book doesn't really give a number for foliate. I kind of wish they did. But that's just something our book doesn't really do. So I try to stick to the book as close as I can, because if a student is trying to read, I don't want too much of what I talk about to not jive with your textbook. But the filiform papilla, they're for friction. Friction is filiform. Like when you lick a sucker and you wear down the sucker, you use the saliva to dissolve it, and you use the filiform to rub that partially dissolved sucker off. That's filiform papilla, the rough, frictiony texture of a tongue. The fungiform have about 5, so think fungiform 5, the fungiform 5. Circumvallate also has, starts with C for century, circumvallate, 100 taste buds each roughly. And then there is actually 12 of these vallate at the back. They form a V shape at the back of the tongue. And the foliate on the lateral margins, they look at the sides of a book, of the folia, the loose leaf, the leaves of a book. That's why they get their name foliate. And so you can actually see the filiform are very for friction. They have these little kind of like tendrils on them that give friction. Fungiform have 5. The foliate on the sides can have a different number. I generally see here 4, and then there's about 100 in a vallate. So I will see what, as I always try to update and improve notes always. I'm always working on them. That's just me. All right. Taste buds are this epithelium of the papilla. They get recessed, and they have the little specialized supporting cells and gustatory cells, and there's somewhere between 40 to 100. Now, some people have less, so they don't taste as sensitively. They don't have as sensitive of a palate as some folks have. Now, because these all are receptors and they have proteins that bind to chemicals, and the proteins are coded for by the DNA, your DNA is different from my DNA. Your taste is different from my taste. Some people, for example, do not like dark roast coffee like what I'm enjoying right now. A nice dark roast, French roast, strongly brewed. I love my dark roast coffees. Some people don't like that. It's a taste thing, and it is due to genetic differences and variation amongst people. So these cells all come in. So basically, again, like let's say you're enjoying a nice fine glass of wine and you sip on the wine and let it go over your tongue to taste it as your body's like, I taste um, hints of butterscotch, plum, and mild spiciness to this. As somebody might be tasting a wine and saying what it is that they taste, that's one of the interesting things about this. As you could see, the gustatory receptor cells will fire and they will send a signal down the neuron, and that will help us determine your body as the peculiar way the chemicals that we taste depolarize these cells in such a way um, they can create a unique signature. And this is how I perceive the differences in the chemical composition of what I'm eating and drinking. So, and you can tell subtle differences. Like, let's say you enjoy a nice Malbec wine and you get a Malbec, let's say you get Don Miguel Gascon. That's my favorite Malbec. Not too expensive, but quite good. And let's say you get a different Malbec another time, and there will be a subtle difference, or you get a different vintage of Don Miguel Gascon. It's a different taste that you get from that. Subtleties, and you can, if you pay attention to it, you can kind of get that. It's like when people taste something like you change brands of peanut butter, okay? There will be subtle differences because of differences in recipe. Now, the how we get this taste to us is this. Now, what I'm going to do is I'm going to talk a little bit about that. I'm going to talk about the tongue here. And what I always like to do, let's just say we have our tongue and we have our pharynx, larynx. Area in the back. Now, on the tongue, we have the front we have this front area, the front 2/3 of the tongue, and then the back third of the tongue, and then we have our pharynx. Now, what is it that nerve is handling that tongue? Well, let's take a look at that together. What we're going to see is, guys, the anterior 2/3 is cranial nerve number 7, CN7. CN7 facial nerve. Sorry, wrong part. Sorry about that. CN7. There we go. I was not paying attention where I was drawing. CN7 monitors the front 2/3 of the tongue. Then we're going to take our back of our tongue and be glossopharyngeal. Glossopharyngeal, and that is CN9. CN9. And what I'll do is, okay, click that, and that is CN9, cranial nerve number 9, glossopharyngeal. And then back here, we're going to deal with our vagus. Our vagus deals like pharynx, larynx, glottis, which is part of the larynx. Our pharynx, larynx area down at the glottis, that's going to be CN10, the vagus, okay? They eventually will all go to the temporal lobe so I can be aware of it, the gustatory lobe. So what's going to happen is all these nerves, they all must combine together, and they're going to go to the temporal lobe for me to be aware of it. Okay, the olfactory cortex, or the gustatory cortex, is found here. Okay. And so all those nerves are going to go in and eventually be able to be perceived. I accidentally said olfactory cortex. I apologize. Now, what can we taste? What are some things that we can actually taste when I eat food? Well, I can taste such things as sweet, like candy, a delicious candy. I can taste salty, like salty potato chips. I can taste sour, like pickles, sour candies. I don't like sour that much. I do enjoy pickles, but I do not like sour candies like atomic warheads. They were very popular when I was a kid. Atomic warheads, I don't like them. Bitter, that would be like some of the bitter things that you find in like horseradish. Umami, that's a savory flavor. You get that from like a nice Parmesan cheese. And water. There is water reception. You can actually, now most of the flavor of the water you drink comes from what's dissolved in it, yes, but there are water receptors. There is a water taste that we know we're getting water. All right. Now, you may notice if you have a stuffy nose, when you guys get a head cold, that your food doesn't taste so good. Your drink, your favorite drink doesn't taste so good. That's because what we actually think of when I say this doesn't taste good is actually flavor. Flavor is the combination of smell and taste. So when you have a stuffy nose, you can't actually do that. Though the gustatory receptors are functioning, you're not getting stuff up to the olfactory receptors, and you're missing part of the dimension of food. This is oftentimes like if you talk about people going to tastings, they do a thing where they like open up their throat and they let the after vapors go up into the nasal cavities to get the full effect of something like a whiskey tasting or something like that. Uh, so. All right, so let's get into the ear, and that's where we're going to spend the rest of our time today. Is the ear? That's the hard part. We're going to talk about the ear is divided into three portions. Now, I'm going to go through this on a little faster pace because we do this in lab as well, and this is more so for edification from lab. So let's start off with the external ear. That's the visible part. It collects sound waves, directs them towards the eardrum or tympanic membrane. The middle ear is in the petrous portion of the temporal bone. It's a chamber inside the skull. It amplifies sound waves, helps us turn the sound waves into louder things to make sure we send it to the inner ear. The inner ear has all the sensory organs for balance or equilibrium and hearing. Now, as we go through that, you can see the external, middle, and inner ear and their compositions. So let's talk about the external ear. The external ear has an auricle, also called pinna. It is to gather sound waves. It's kind of like a little bit of a funnel for sound, and it takes and gathers sound waves, and those sound waves get directed into the external auditory canal or external acoustic meatus, and that meatus takes it to the tympanic membrane or eardrum. All along the external acoustic meatus, there are going to be glands called ceruminous glands that make earwax or cerumen. Then there's the tympanic membrane at the end, and that's what vibrates to the sound waves. This is the structure that says you are now no longer external ear. Once you cross this and you go to the other side, you're now middle ear. It is like the border between the external ear and the middle ear. Now, in the middle ear, you're going to see our tympanic membrane separates that, our eardrum, and behind it, there is a chamber here called the tympanic cavity. And the tympanic cavity is our resonating chamber. It's the cavity in the middle ear. And attached to this is our pharyngotympanic tube. Why it's called pharyngotympanic tube is one end is attached to the pharynx. The other end is attached to the tympanic cavity, and this allows us to connect the tympanic cavity to the pharynx. Now, inside the tympanic cavity, there are three bones, the malleus, the incus, and the stapes. The malleus, which is attached to the tympanic membrane here, will vibrate. When the tympanic membrane vibrates the sound waves, you will see the malleus will rattle into the incus. The incus will rattle into the stapes, and the stapes will do its tulip plunger thing on the inner ear. Now, there are two muscles, and if I zoom in here, I could show you these two muscles. One muscle here called the tensor tympani, and a muscle here called the stapedius. These two muscles reduce the impact of sound. One is on the malleus and pulls to tighten the tympanic membrane. It inserts on the malleus, reduces the movement of malleus, and thereby also reducing the movement of tympanic membrane. It's innervated by the trigeminal. Think tensor tympani trigeminal. Stapedius is on the stapes, reduces movement of stapes. The stapes is moving up and down like a toilet plunger in the bottom of a toilet, and that will stimulate the inner ear. If you could reduce the movement of these bones, you reduce the perception of sound. See, some people get a condition where their ear bones, inner ear bones, ossify, and they can't hear well. This is what happens. The stapedius muscle and tensor tympani reduce the movement of these bones to try to make it harder to hear, for example, when exposed to loud noises. Now, the facial nerve does stapedius. I always think if someone's talking too loud, you want them to stop talking, you might say STFU, stapedius, STF, facial nerve. So think STFU, which we all know, shut the frick up. I'm just going to be nice with that. Okay. So, all right. Now, the auditoryossicles, what they are, you can see them here. Malleus and stapes are very small bones in the tympanic cavity. They rattle together to transmit the sound waves vibrating on the tympanic membrane to ultimately to take those as vibrations and pressure waves into the inner ear for sound. All right. Now, how does this happen? Now, our inner ear has these two labyrinths, two portions called labyrinths. Now, the inner ear is got the receptors for hearing and balance. And so what we got to do is take fluids of the inner ear and slosh it around to make that stimulate receptors. Now, we have two portions, what is called a hardened bony labyrinth and a softer membranous labyrinth. So the bony labyrinth coats the outside in osseous material and the membranous labyrinth inside in a soft membrane. Okay? Now, let's talk about our bony labyrinth. Where do you find bony labyrinth? You find it in three places, but it is bony tissue and it covers and protects the membranous labyrinth. It's like the home of the membranous labyrinth. Think about the membranous labyrinth is something soft that lives inside of a house. It lives in a house of bone. Now, the three places, the semicircular canals. Canals hold the ducts. Canals surround the ducts. The vestibule, it will house and hold the utricle and saccule. And the cochlea holds the cochlear duct. Looks like a snail shell. Now, let's take a look at this and talk about it and talk about cochlea. And let's talk about we'll use cochlea as an example in my cochlea unroll drawing. And let's say I unroll a cochlea. We had you guys draw this in study group for those who came Friday. And on Friday, I promised I would redo my my drawing. I would redo my recording here, and I'm doing it just like I promised. And so let's kind of take a look at this. All right. So real quick, what do we have here? I have a bony labyrinth. And I have a membranous labyrinth. These two labyrinths, okay? Now, what's going to happen is we are going to have an opening here and an opening here. These are the windows. You have a round window down here, and you have an oval window up here. And remember, the oval window has the stapes, And the stapes does its toilet plunger thing on the oval window. Okay? Okay. Now, what I want to help you guys understand here is the organization of that. Okay? Now, the oval window vibrates when exposed to movement of the stapes, which creates pressure waves in what is called the scala vestibule, which I am just going to refer to as the vestibular duct. We're going to call this the vestibular duct. Sorry, I'm about to misspell vestibular. I'm going to call it the vestibular duct. So what happens, this creates pressure waves in the vestibular duct. Okay. Now, let's say these pressure waves, what they can do is they can pass by the middle duct here, and there is what's called a tympanic duct. I'm going to put duct underneath that. Okay, there is a tympanic duct. Okay. Now, the tympanic duct, what happens is these pressure waves that are in here, they get dissipated by that to prevent auditory echo. Okay. Now, we're going to pick back up with this in a minute. And what we're going to see is this is our cochlear duct. Okay. Our cochlear duct. Now, what I want to do is talk about that in just a second. Now, the membranous labyrinth, that is inside, and the cochlear duct is the membranous labyrinth in the cochlea. Now, what you have is in the membranous labyrinth, these are filled with endolymph. So what we're going to do is, I forgot to add this to my lecture, to my drawing, is there is the bony labyrinth has what is called perilymph around it, inside of it. I think I'm trying to remember where I actually discussed it. I know I talked about it having perilymph somewhere. Yes, the fluid between the bony and membranous labyrinth. So let's add that there. Let's make sure we know that perilymph is found here. Okay, perilymph. Now, inside of this is endolymph. Okay, we have endolymph inside there. Now, what happens is these hollow chambers of fluid are going to have pressure waves go through them, and we're going to distort the membranous labyrinth, and that's going to help us to begin to either hear or feel balance. Now, to do balance, we have what's called the vestibular complex, which is the equilibrium receptors that is made up of the vestibule and the semicircular canals. Now, the vestibule, remember, is the house for the utricle and the saccule to live in. The utricle and saccule are the membranous labyrinth that lives inside the vestibule. The vestibule is the bony labyrinth that is inhabited by the utricle and the saccule. The semicircular ducts, they live in the semicircular canals. Duct is soft, canal is hard. The cochlear duct is the membranous labyrinth in the cochlea, and this does sound we're going to talk about, okay? Now, let's get in and talk about the utricle and saccule a little bit. Now, the utricle and saccule, what they have inside of them is a structure called a macula. These are for gravity and linear acceleration. They contain a macula. The macula is a specialized sensory organ for gravity and linear acceleration. So this tells you up from down and forwards from backwards. Which direction am I going in, and am I moving up and down? This tells you which way's up and which way am I going. Okay, gravity and linear acceleration. To do this, we have the macula, which is a cluster of hair cells. Here you can see in it all these hair cells. These Hair cells are specialized, remember, mechanoceptors. Mechanoceptors, remember, distort. When things bend them, they depolarize or hyperpolarize when cell membrane distorts, sending or not sending signals. Now, kinocilium. There is on them a long, single, long kinocilium. There's one of them. The kinocilium, the other cilia called stereocilia and stuff like that, they bend either towards the kinocilium or away from the kinocilium. And depending on whether they move towards or away from it, tells us what direction we're going in, okay? Okay, sorry, I had to take I'm just now getting over COVID, so I have to keep taking a sip every now and again. These little peachy supporting cells hold these hair cells up into this gelatinous membrane called an otolithic membrane, which covers the hair cells. The hair's little cilia, stereocilia and kinocilium, are covered in this gelatinous material, and then on top of the gelatinous material are what is called an statoconia, which ends up making an otolith. Otoliths are the combination with the membrane and the statoconia. The statoconia cells, the otolithic guys here, they bend and distort this, and they help accentuate the movement of the otolithic membrane of the gelatinous material. All right. Now, something I always test on in lecture and lab is the semicircular ducts and what kind of movement they perceive. So semicircular canals surround the ducts. The ducts do rotational motion. Like if I shake my head up and down in a rotational movement, like doing front flips and back flips, going yes, that would be detected by the anterior semicircular duct. That does vertical head movement, or me doing front flips, back flips. The posterior does head tilt. That would be like cartwheels, or me going, I don't know, bobbing my head from side to side. Lateral does rotational movement, like spinning like a top, or like when you put your head on the back of a bat handle and then spin around in circles. That's lateral. That does rotational movements, like I shook my head no. Okay? You will get tested on this in lecture and in lab. All right. Now, let's talk about how we do the rotation. How is it actually detected? Well, if you look right here, at the base of each semicircular canal, here, here's the base, here's the base, here's the base. There are these organs here. These organs, look inside them there, and you see this little thing right here. This is called an ampulla. The ampulla is what is at the base of each semicircular canal. The base of each semicircular canal has an ampulla and a large area, and there's a special receptor in there. This ampulla has a crista, which is this little hump of cells, supporting cells, that contain embedded in them, sorry, I didn't mean to click, embedded in them, these little hair cells. And these hair cells send their cilia up, and then we stick a little jelly filled little capsule on top of it called a copula. And when this copula bends like a light switch, this way or that way, it distorts the cilia. And when the cilia of the hair cells get distorted by movement of the copula, they move the copula. It's like a light switch being flipped on or off. We alter that movement of hair cells, which tells me what direction I'm moving in. It alters the rate of neurotransmitter release, as I'm going to talk about a little of the physiology of hair cells in a moment. Now, these hair cells, external forces like movement of the macula's gelatinous membrane, movement of an copula, will bend the hair cells that are in the gelatinous material. When the hair cells bend, depending on one direction, they're stimulated. If they are towards the kinocilium, if they are going away from the kinocilium, they're inhibited. It's like the on-off switch. on your lights. So what happens is distortions alter the rate of neurotransmitter release, and this tells the brain which direction I'm moving in. Now, if I happen to be in the vestibule, I'll do gravity and acceleration, things like up from down, and I'm moving forwards or backwards. Semicircular ducts do things like rotational movements like yes, no, and head tilt. Cochlea, its hair cells, will give us information about sound. Depending on where you are, you will get unique subsets of information because of the structure that you're in. Its hair cells are tuned to those types of pressure waves and movements, depending on the organ that has them. If it's in a macula, if it's in an ampulla, or if it's in an organ of corti. Okay, that's how come is, remember, labeled line principle. That went back, I taught you guys labeled line principle, my A&P 1s back, but if you get, most of you guys didn't have me for A&P 1. And I realize I'm a very different instructor. I do go into the details and really talk about how things work. I'm a very much how does it work kind of guy. And I know that's because I really come from academic disciplines, not so much as like I was not a person who did practice. I did academics, and that means I geek out over details. Now, the hair cells that are present, what they're going to do is they're going to, in the macula, they're going to be about gravitational pull and linear acceleration. Now, for example, to do this, if you're a macula and you're in the utricle, you do horizontal movements. That would be like forwards, backwards, okay? That would be linear acceleration. So when my body moves, this tells me and stimulates the hair cells. So macula, the saccule, does vertical movements, gravity. I will test on one of those two, for sure. Okay, one of those two for sure is what does the macula, the utricle do? What does the macula, the saccule do? So then if you lay down horizontally when you're asleep, you might drool, you trickle. You trickle drool out your mouth when you lay down horizontally. Saccule, if you got a sack of money, you want to keep it vertical so your money doesn't fall out. Saccule does vertical movement. Okay. All right, now let's talk about the equilibrium pathway. The equilibrium pathway, I like to talk about it because I do want my students to understand that the vestibulocochlear nerve had two branches. It had a vestibular branch and a cochlear branch. The vestibular branch of cranial nerve number 8 takes this information to three places: the cerebellum, the midbrain, and the cerebral cortex. Now, the cerebral cortex lets me know where I am consciously. The cerebellum will help use my balance information to adjust the activities of the somatic motor pathways. And so will the midbrain also use it to affect other things. For example, eye movements along with ear movement, things like that. And they alter muscle activities, okay, of the body. All right, so we've done the cochlea. We talked a little bit about it when we did our drawing of the cochlea. And we talked about that there are a scala vestibule or vestibular duct, a scala media or cochlear duct, and a scala tympani or tympanic duct. And together, these three layers in the cochlea, imagine you roll it up like a cinnamon roll, and it would look like that, okay, as a spiral. Now, what's going to happen is, guys, we have some other things that we talk about, like the layers of this duct here and here. There will be a basilar membrane and a vestibular membrane. And together, the vestibular membrane makes the roof. I like to think about a roof is like if you think about the roof of a building as an upside down V, okay? Think V vestibular, okay? That's how I like to remember vestibular duct. The vestibular membrane is the roof of the cochlea, is that upside down V looks like a roof of a house. Okay, that's how I remember it. Okay, and I like to help students remember things sometimes, of course, all the time, as much as I possibly can, not just sometimes. All right. But I like to give you the little tips and tricks that I have. Now, the cochlear duct scala media is responsible for hearing. It's found in the cochlea only, and it's between the scala tympani and scala vestibule, or the vestibular duct and the tympanic duct, which I prefer to say that. That's how I was taught it, not using the word scala tympani and scala vestibule. I was taught it as tympanic duct and vestibular duct. To separate your vestibular duct from the cochlear duct, you have vestibular membrane. To separate the cochlear duct from the tympanic duct, you have the basilar membrane. The basilar membrane holds the organ of Corti, or spiral organ. Now, I usually call it organ of Corti. That was the first word that imprinted on me as a student, but spiral organ is more common now. The spiral organ, because it spirals throughout the cochlea, it is found with a bunch of hair cells that are for sound. These hair cells are sensitive to movement. In them, their hair cells only have stereocilia, no kinocilium. And they are distorted when you move. The tectorial membrane vibrates, which moves the hair cells and stimulates them. The spiral organ kind of works like an old-timey telegraph. And if you think about the old telegraphs, the old-timey telegraphs, when the tectorial membrane beats against the hair cells are distorted and moved. That will alter the related neurotransmitter release, which will begin to create signals that we will perceive as sound. The tectorial membrane covers those stereocilia and distorts them, and as pressure waves go up and down, you will start to see the movement of the tectorial membrane will then beat against and vibrate against the stereocilia of hair cells, creating a rhythmic pattern of stimulation that will create the signals we know as hearing. The scala tympani will then take the round window and use that to dissipate and conduct the pressure waves out and prevent the auditory echo. Remember, the scala tympani and the scala vestibule have perilymph in it. So if we look at it like this, your cochlea, which is rounded around, we have our vestibular duct, our cochlear duct, and our tympanic duct. The organ of Corti, or spiral organ, is located between, and it spirals, as you can see, throughout the entire cochlea. And right here, as it is with hair cells, there are hair cells, and then there's a tectorial membrane over it. And it works like an old-timey telegraph. Okay. Now, how do we actually use sound to create unique signals? How do we do it? Well, sound has characteristics to it. Sound, just like light, has unique properties according to whatever made it. Sound has waves to it. And because it has waves, we have wavelength. And wavelength can be measured and give us characteristics of that sound and unique properties. So what is a wavelength? If I made a sound like "ooh" or "ee, ee, ee," and I made sounds, what I would be able to do is measure the wavelengths of these sounds. And when I have the little high spots on it, a crest, and I have another crest, and I measure the distance between these two crests, I have what is called a wavelength. Now there's long wavelength, like "ooh," and short wavelength, "ee, ee, ee." And these are different characteristics of sound. These are different sounds have different wavelengths, okay? So, like, for example, pitch and wavelength pitch is like your perception of a wavelength. Now, wavelength, if I have, for example, here, let's say that I measured the same amount of time, okay? Let's say both of these were measured in the same length of time. Here I have a bunch of waves in that same amount of time. Here I only have 1.5. So this would be low frequency. This would be high frequency. Okay, for example. And what happens is frequency is something, when I measure wavelength, I get something in what we call frequency, which we measure in cycles. Now, let's say I measure this in 1 minute. That would be a hertz. That would be how many hertz? Hertz, for example, is a measurement we use for sound, for how many cycles in a minute, which cycles is a measurement of wavelength. And how many wavelengths are there in 1 minute gives us an idea of the frequency. Pitch is our perception of that. Now, case in point, some people, you don't like their voice. You may go in there and say, I can't stand Justin Bieber. He's so pitchy. He's high-pitched and annoying, and I can't stand it. Or it's like some musicians, you can't stand it. Some musicians you really like. Now, like some musicians I like, I might be listening to a musician with a very deep, gravelly voice, and some people can't stand it. They're like, oh, it's off-putting. And things like that. Now, amplitude is how much energy there is. Now, for example, with amplitude, we can measure that by getting an idea of the top and the bottom of those things. Okay, this would be like my amplitude. Right here would say, okay, this is amplitude. If I yelled really loud, or if I was really quiet, okay, you get different amplitudes. And amplitude is measured in decibels. Like a jackhammer is about 500 decibels. So sound itself has these properties to it. And because there's unique properties to sound waves, our ears can take these properties and alter the neurological signals it sends according to it to tell us about what it is that we're hearing. And how does it do it? Well, here you can see a sound wave. We vibrate and we create a wavelength. The wavelengths and the amplitudes, exactly what I was showing you guys when I drew it, okay? Now, what happens, and I could show you, for example, here, is to determine it, is like if you're very high-pitched, high frequency, this is real high frequency, this is like Justin Bieber squealing, baby, 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 oh baby. I can't even get that high. And then down here, this might be like Coulter Wall, like, sing, listen, my sense catches up with me. And this might be somebody in the middle, just, like down here, you've got like Steven Tyler, dream on, dream on. You got that. Sorry, I'll be a little bit interesting today, I guess. That's what we mean. This is very like low hertz. This would be like very deeper voices. They penetrate into the cochlea further and travel further. Where high pitched, like Justin Bieber squealing, baby, 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 oh baby, doesn't tend to penetrate the cochlea as deep. Okay, so how do we hear? Well, let's kind of take it. I'm going to do it like this. I like to do this. So right here we have our tympanic membrane. Sound wave with all of its properties, wavelength and amplitude, hit this. And based on its unique sound wave signature, amplitude and wavelength, it distorts it in a unique way, which makes the malleus, the incus, and then the stapes vibrate. Now, the stapes is sitting here on the oval window. And when that pushes in the oval window, it causes pressure in the scala vestibule, which will then distort the cochlear duct and pass it off to the scala tympani. Notice as this goes in, that goes out. Now, on our cochlear duct is our beautiful hair cells of our on our tectorial membrane and basilar membrane, and they vibrate together. And depending on the region where we actually stimulate the cochlea tells us about the intensity, information about the region and the intensity get. Now, high frequency doesn't penetrate. The region is very close to the entrance of the ear. Very low, long wavelength, low, deep tones, like somebody that's singing a song. They got a real deep, gravelly voice. Somebody like Coulter Wall or somebody like that with that deep somebody's got gravelly. I like a lot of different music. Americana is some of the music I like. I also like a lot of other things, and I still listen to some of the songs, some music I liked from the '90s as well, every now and again. But I like a lot of new modern bands as well. Just typically people who are playing some things I like. I don't like everything, but that's okay. There's some kind of modern bands that I enjoy. And what we would look at, so sound gets to the tympanic membrane. We cause displacement of the auditoryossicles. We move the stapes at the oval window, which we slosh and create pressure waves in the vestibular duct. Those pressure waves distort the basilar membrane, which will cause the hair cells to collide with tectorial membrane, which will then be able to tell us about where on the cochlear duct do we stimulate it, which hair cells, and how intense is it. If you slap a bunch of hair cells, then it's very intense. And eventually it goes to vestibulocochlear nerve. It goes out the cochlear branch to vestibulocochlear nerve, cranial nerve number 8. Eventually got to get to where you can hear it. So sound waves, pressure comes in, hits and vibrates the tympanic membrane. We rattle the malleus, the incus, and the stapes. The stapes does its tulip plunger thing on the vestibular duct. Pressure waves generated in vestibular duct will go past the cochlear duct on their way to the tympanic duct to the round window to be dissipated. And when that happens, what you're going to do is you're going to take the organ of quarti here and stimulate it, and the hair cells will fire, and it will send that signal out to the cochlear nerve to the brain. So we said frequency. For example, determine on what part of the cochlea. Here, this is high frequency, very high pitched, like, dream on, dream on. And then over here it's like, hey, darling, running through the trees, honey. Very deep. I can make my voice go. I can't do too high pitch. I've been told my voice is high pitched and annoying. Please don't say that on my IDEAs. That made me feel bad. Somebody wrote that once. The intensity is how many hair cells. So this tells me how loud. So like if I'm stimulating right here, this would be like middle of the road, like just a normal voice. Right here, really deep voice. Somebody who's really got the deep. This is a really deep voice, darling. How are you doing? And then very high pitched voice right here. Now, like bird songs, a lot of people. Because it's very hard to, you lose the high frequency very early. High pitch gets lost very early. As we get older, we tend to lose the high frequency, high pitch sounds, those ranges first. They tend to be the ones that are hardest for us to keep. So young people can really hear that very well. Now, auditory pathway. Let's talk about that. Basically, what happens when my hair cells and my cochlea produce things, depending on like, is it low frequency or high frequency? High frequency. This is Justin Bieber, and this is somebody with really deep voice. I'm trying to think of other singers who have deep voices, other than somebody like Coulter Wall or something. When I was getting my um, when I was getting my root canal done, Coulter Wall was on their playlist playing, and the dentist was just excited that I knew who Coulter Wall was because she's like, I picked that playlist. Nobody knows who he is. I was like, I enjoy his music. But I also do like Americana sometimes. It's not the only thing I listen to. I also like Celtic punk, and I got a wide range. I love the Avett Brothers. And anyway, sorry, I'm talking about music, getting a little personal today. Okay, so once we get that information, we go to the cochlear nerve, vestibular-cochlear nerve. It's got a branch called the cochlear branch. It goes out, CN8. It ultimately goes to the pons, which there's a nucleus in the pons that it goes to. And what we basically talk about is we'll send it up. Now, the thing is, in the midbrain, it goes up. Some crosses over, some remains uncrossed. Some crosses over, some remains uncrossed. It goes up to the thalamus and then out to the appropriate part of the brain. Now, here's the weird thing. If it's high frequency, it's supposed to go to a specific. If it's low frequency, if it's middle, it's supposed to go to another area. Now, it may not go to the right spot of that. Now, it goes through, when it goes through the thalamus, it goes to the medial geniculate nucleus. The medial geniculate nucleus. To hear me, it's medial geniculate nucleus, okay? Know this pathway. I will ask it, and I will ask parts. Like, I might ask you, like, I might try to trick you up on lateral geniculate nucleus, which is visual pathway, to look at me, you need lateral geniculate nucleus. To hear me, you need medial geniculate nucleus. All right, this is where we're going to end it. And the first lecture. And then I'll email you guys, let you know I sent it out. It'll be in UJEF first. Of course, if you're listening to it, then I guess that makes no sense. I'll email you guys. Guys, thank you. But this will be up on YouTube. And thank you guys so much for watching the first half of this lecture. I hope you guys learned a lot. Guys, thank you. And I look forward to seeing you in the next one.